Hello and welcome to our first offline lecture slash answer key. So what I'm gonna do for these videos is that I'm gonna just post up a verbal explanation to the answers for the questions I posted for today's lecture. So the questions that I posted, they're your reference guide to what material to really focus on, and they're also a way to test your understanding of the material. So when going through the video lectures I have in each folder, watch those, then go to the questions and see, did I understand these questions? If not, go back to the videos and see if you can answer them. So I really want this video to serve as like after you've tried, after you struggled a little bit, if you're still not sure, you can come here to see what's going on. If you have any questions, please don't hesitate to email me. I can answer any questions over email, and we can also set up a video chat so we can talk face to face like that if you prefer. But right, let's get into it. So our first question is, what determines the half-life of cytoplasmic proteins? And that's usually their N-terminus residues. Depending on what residue you have, you can have a half-life of 2 to 3 minutes, or you can have a half-life of 10 to 20 hours, and that's due to interacting with the E3 enzyme of our ubiquitin pathway. And also pest sequences can cause ubiquitination as well. So lysozyme is where we destroy proteins. What happens if our lysozyme splits open? Would we just degrade all of our proteins inside of our cell? And the answer is no. So lysozyme only really works, or the proteins inside lysozyme only really work if the pH is roughly 5. So if you have your lysozyme bursting open, the cytoplasm is roughly 7.2. And so your proteins inside your lysozyme would become inactive, and you wouldn't eat your own proteins. What are the steps for ubiquitination, and what are the roles of E1, E2, E3? So it is a 3-step process. First, E1 uses ATP to attack ubiquitin to itself. Then E1 passes this ubiquitin over to E2. E2 then passes this ubiquitin over to the protein we want to become ubiquitinated with the help of E3. So E3 is actually the protein that is transferring from E2 to the protein. We are always attaching ubiquitin onto a lysine residue of whatever protein we are ubiquitinating, and we will form this isopeptide bond, which is a peptide bond but on the side chain instead of the backbone, and then we can polyubiquitinate, keep adding ubiquitin, and ubiquitin. What destroys ubiquitinated proteins, and basically how does it work? So the 26S proteasome is the superstructure that is going to destroy our ubiquitinated proteins. Basically, how it works is that the 19S will recognize ubiquitinated proteins. It will unfold them, and then it'll go into the 20S to chop them up. And it's only 3 beta subunits, beta 1, beta 2, beta 5, that's doing the chopping. Beta 1 after acidic residues, beta 2 after basic, beta 5 after hydrophobic residues. Question 5. So what happens to the ammonia and carbon skeleton of proteins of amino acids when they are degraded? See that my text cover this a little bit, but the amino acid, the ammonia, will usually go to the urea cycle. The carbon skeleton can be used in a lot of places. It can be broken down carbon dioxide, can be made into glucose, can be made in acetyl-CoA, or it can be made into ketone bodies. So there are a lot of ways that we can use the carbon skeleton of our amino acid. What is transamination, and how is transamination and the citric acid cycle related? So transamination is simply moving the amino group of amino acid to something else. So for general amino acids, it's moving it to alpha-ketoglutarate to make glutamate in an alpha-keto acid. If you have glutamate, you can move that to oxaloacetate to make alpha-ketoglutarate back in aspartate. So it's the moving of that NH3 group. How is it related to the citric acid cycle? Well, we're using alpha-ketoglutarate and oxaloacetate. So these are citric acid cycle intermediates, as we know by now. So this pathway is connected. So the urea cycle and the citric acid cycle can be connected through this process of transamination. What's the cofactor required for transamination? That will be PLP. That is derived from vitamin B6. And as I said in the questions, don't really worry about the mechanism of how PLP works. Mainly want to know PLP is our cofactor for transamination. What inhibits the oxidative deamination of glutamate, and what activates? So for inhibiting the oxidative deamination of glutamine, that's going to be inhibited by GTP and NADH. So the transamination or oxidative deamination of glutamate will remake alpha-ketoglutarate, so we can continue to break down amino acids. If you have a lot of energy, then we don't really need that to happen, so we can just stop taking this glutamate and to make alpha-ketoglutarate. Alpha-ketoglutarate is also in the citric acid cycle, so it could potentially be used for that as well. What's it activated by? ADP and NAD+. Again, alpha-ketoglutarate, we can use that to break down amino acids and go and use the amino acid carboskeletons for energy. So if we have low energy, as ADP and NAD+ is saying, well, start breaking down that glutamate so we can start making alpha-ketoglutarate. And that alpha-ketoglutarate can also go through the citric acid cycle and give us energy. So really not much of a surprise based on what we've already talked about this whole semester, where you're high in energy while you have GTP or ATP, NADH, so don't make more energy. You're low in energy, so you have stuff like ADP and NAD+, start doing reactions that make your energy. What three molecules do organisms use to excrete excess nitrogen? Ammonia, urea, uric acid. So if you're aquatic, you can just get rid of ammonia. If you're not surrounded by water all the time, you're either going to break it down in urea like we do, or uric acid like birds and terrestrial reptiles do. Urea cycle. Not much I can say about this. This is going to be our big memorization pathway for this section. Shouldn't come as a surprise to you, and from talking to many of you, it seems like you have mechanisms to already memorize these pathways efficiently, so another one shouldn't be too bad. Interestingly enough, this will span the mitochondria and the cytosol, so we're going in and out of different organelles for this pathway. How is the urea cycle regulated? So on a previous slide I talked about CPS1, so make sure you know what CPS1 is and why it's important. Just shorthand, it's the first step of the urea cycle. It's not actually in the cycle, but it's similar to PDH in the citric acid cycle, in that we're getting our molecule ready to enter the urea cycle. It's the slowest step. It's the rate limiting step. So the regulation of this is that CPS1 is turned on by the molecule N-acetylglutamate, as shown here. And this metabolite is made from glutamate in acetyl-CoA by the enzyme N-acetylglutamate synthase. So as we break down amino acids, the concentration of glutamate will increase. The more glutamate we have, the more acetylglutamate synthase we have, which will mean CPS1 will turn on and will run through the urea cycle, which again makes sense. If we're breaking down a lot of amino acids, we're going to have a lot of ammonia breaking or building up. We need to get rid of that ammonia. It is toxic to us, and so we need to turn it into urea so we can excrete it out of our body. So make sure you turn on the urea cycle if you're breaking down amino acids. And that is the answers to those questions. I didn't go in as much as depth as I would during a normal class period, because I do that in the actual video lectures, and you should have those available to you at home while you're answering the question for while you're answering each question. But again, if you have additional questions or you want to set up a time to talk about anything, again, please don't hesitate to reach out. Otherwise, I will see you in the next video.